last week we looked at protozoa, but before that time we'd looked at bacteria. Now this week we're focusing on the fungus kingdom. We're focusing on other eukaryotic organisms. We're looking at the two different types of eukaryotic fungi. One will be the yeast category and the other one is going to be the mold category. So for the yeast lab, we're going to look at yeast in a couple of ways. One, we're going to look at their structure. Now recall with bacteria, we had different shapes. We have a coccus, we had the bacillus, we had the spirillum, and then we had the different arrangements. Now with yeast, because they are eukaryotic, they're going to be larger than the bacteria. But growing on a plate, the colonies might look somewhat similar, probably a little bit bigger, but you're going to see the same round colonies that you might have seen with some of the bacterial species. But with these eukaryotic organisms, when we go to look at them under the microscope, you're never going to have to do any higher than 40 power or 40 objective because they're not small like bacteria, so you're not going to need the oil immersion. And also when we look at them, we're going to look at their shape. Now they don't have, you're not going to use bacillus and coccus and things like that for the shape. You're going to use general terms like round, oval, elongated, okay, when we look at the yeast. So when you go to look at them under the wet mount, keep those things in mind. The other thing that you're going to look at is how they divide. So yeast basically divide in two different ways. One is they divide by budding. And budding is where a cell will have a small outgrowth and then that outgrowth will pop off, okay? So what you'll see is a larger cell with a small one coming off of it, or you might see the larger one with the smaller one already off. It's not going to be symmetrical, okay? The other type of yeast is the fission yeast. And those are going to divide by elongating, so they're going to look longer, and then they're going to pinch off. So the two daughter cells are going to be symmetrical, okay? So that's how we're going to divide them, is based on their symmetry, how they divide, and based on their shape, the oval, the round, and elongated, okay? And also you should be able to see a nucleus in these cells because they are eukaryotic. The other thing is when we talk about eukaryotic organisms and we talk about things that are a fungus, you want to keep the fungus characteristics in mind. They all do decomposing. They are all made of eukaryotic cells, but the difference between a yeast and a mold is the fact that the yeast are single-celled and the mold are multicellular. Also, the molds will produce the hyphae in the filaments and the yeast will not, except for the Candida albicans. It's called pseudo hyphae, where they have little extensions and then the buds off of them. They're not true filaments, that's why they're called pseudo hyphae. Okay, so when looking at this yeast lab, the first thing that we want to talk about is one of the species of bacteria we do not have. The intestinales species is one that they actually do not make or we cannot get a hold of, so it's not one that we're going to use. We're only going to use the Saccharomyces cerevisiae, which would be better known to you as baker's yeast. We're going to use Candida albicans, which is the cause of yeast infections. We're going to use Rhodo, Rula, Rubra, and Rubra means red, so these actually have a pink color to them. And the last one is the Schistosaccharomyces octosporus, and this one is a more white in color and it can look a little bit fuzzier than the regular yeast does. Okay, so we're going to look at those four. We do not have the fifth one. Okay, therefore when you go to do your tubes, the lab manual says 5 sets of tubes. We're doing 4 sets, and you have 4 different sugars. So we have glucose, sucrose, maltose, and lactose, because yeast can be identified on the type of sugar that they break down. So for example, a yeast might break down sucrose and that's it. They can't do any of the disaccharides or any of the larger sugars, okay? Some will prefer milk sugar like lactose. Some will prefer a different sugar. So just like we could identify bacteria based on their biochemical tests, we're going to do the same thing with yeast. But here we're just looking at specific types of sugars that they ferment. Just like with the fermentation test before, the tube that you're starting with has a Durham tube in it that you're going to look for gas in. Now back when we did the fermentation of bacteria, we had phenol red as our pH indicator. In this case, we're going to have bromocresol purple. So you can see it's a really pretty purple color. It does still have the durum tube inside of it. Once again, like you did before, notice if there's a bubble in the durum tube before you ever use it, and you need to make note of that bubble. But just like with the other fermentation tests, this bromocresol purple will also turn yellow in the presence of acid. So fermentation of yeast in yeast is very similar to fermentation in bacteria. You get the same types of waste products, okay? So the other thing that you're gonna have is 4 sterile saline, so it's kind of saltwater solution. And so for one part, well, before you start putting them into tubes, the first thing that you're gonna do is make a wet mount. And in this case, you're gonna be using a stain called lactophenol cotton blue. It's in these black containers, and it's a little dropper bottle. Do be aware that this has phenol in it, so it's gonna smell a little bit like the carbonyl fusion that we used in the acid fast stain. When you do the wet mount for it, we're gonna be taking from a solid medium. So instead of doing water, you're gonna do a drop. It says a couple of drops. I think 1 drop is always sufficient. So a drop of this, and then you're gonna use your sterile technique. You're using your inoculating loop and your Bunsen burner, and you're gonna take a loop full of the specimen. Now the specimen, they're gonna be on plates, okay? So when you take your loop full from the plate, make sure that you're gentle and you don't gouge up any agar. Just take you need just 1 small colony, not even a whole colony to make a wet mount, because if you get too much in there, it's going to end up being all clumped together and you're not going to be able to see the cells. So like we've done before, less is more. Just basically touch the colony and then spread it into the lactophenol cotton blue, and then put your cover slip on. It's a wet mount. You're not making an actual smear, okay? So before you do anything else, you're gonna look at them under the microscope. So each of the 4 species will be on a separate plate, and you're gonna look at their shape and whatever structure you can see. Then you're going to, using the loop, you're gonna be sterilizing it like you've done before, and you're gonna take the 4 specimens from the plates and putting them into the appropriate glucose, maltose, sucrose, and lactose tubes, okay? Once again, you don't need a lot. You don't need to gouge into the agar. 1 colony is fine, okay? The last procedure that you're gonna do is we're gonna be looking to see if there's any kind of spores or sexual reproduction that goes on with these yeast. So you have 2 plates of the glucose acetate agar, and it's kind of a spore enhancing agar. And so because we have 4 species in 2 plates, you're gonna divide each plate in half and write the name of the species on either side of the plate. And then you're going to take a loop full of the bacteria from the original plate. In this case, get a little bit more. Once again, don't gouge into the agar, and you don't need the whole plate. A couple of colonies is fine. But you're gonna take it from the plate, and you're gonna put it into the sterile water, and then you're gonna give it a little bit of swirl, so you see cloudiness and you see the turbidity, and you see that it is evenly distributed. And then you're gonna have, and I forgot to bring them out here, you're gonna have some sterile plastic transfer pipettes. So they're in the plastic wrap, and they're the ones with the bulbs. And so basically from the sterile water that you took from the original stock plate, you're gonna put a drop, just 1 little tiny drop of the saline solution with the culture of the yeast on the appropriate side of the plate. Now you want to let these sit for about 5 minutes right side up to allow the solution to go into the plate, to diffuse into the agar, and then you are going to flip them upside down in the incubator like we've always done. Now all of these, because they are fungi, they will want to be at 25 degrees instead of 37. So all of these we're going to put in the 25-degree incubator. And it says for the fermentation tubes that you're going to go for 4 to 5 days. We're going to basically do this all for a week. So we'll start this Tuesday, this week, and then we'll do it 1 week later. We'll look at the results. And The the plates say to go for 7 days, so since we're doing it all for 7, we'll just keep it consistent. On the second last lab period, a week later, you're going to be looking for fermentation, acid and gas, just like we did before. And then on the plates, you want to visually look to see if there's any sporulation going on. You can do that with a hand lens, like a little magnifying lens, but also we have the dissecting microscopes that you can use, which we will use to look at the molds. Okay, and so you'll know how to use those. You're also going to make a wet mount from the plates in that second week, and you're going to do it this time with iodine water solution instead of the lactophenol cotton blue. When I say that 3 times fast. Same thing, you just need to put a drop of the iodine on the slide. Then you get a small sample of your culture, put it on the slide, put on your cover slip to make a wet mount. So this is the lab for the yeast characteristics in reproduction.